Opportunity to chair this very exciting session, Molcom for therapeutic local drug delivery in the oncology case. However, because I'm always disruptive, I have opted to ask, my valued, speakers here, not to focus on any data, they may have, but rather introduce ways they could use their own discipline, their own area of expertise in resolving and offering solutions in this particular area of major interest, drug delivery and especially cancer. I'm very proud to announce here Professor Tanashi Takano, who came all the way from Japan, Osaka University, a renowned expert in the field of molecular communications. And what he's going to present today is mobile molecular communication networks for targeted drug delivery.
Okay.
Welcome.
Thank you very much. So I'm talking about mobile molecular communication networks for drug delivery applications. So this is molecular communication. You have been talking about this from yesterday. I wasn't here yesterday. So in molecular communication, we use chemical signals or molecules for communication among the bionanomachines. They have senders and the receivers, and they communicate by propagating molecules in the environment. And the senders and the receivers, the communication devices for molecular communication, bionanomachines, biology or chemistry-based devices. And these are bionanomachines, very small scale, nano scale to micro scale. But in most of my paper, in past work, we're talking about micro scale bionanomachine. It sounds a little strange, but we have been using this term bionanomachine, so we keep using this term. But we often assume that the bionanomachines are based on the macro scale devices. And they are capable of sensing, processing, releasing chemical signals. And the examples of bionanomachines are biological cells. They are naturally occurring bionanomachines or genetically engineered bionanomachine cells, or it could be synthesized from scratch using chemistry technique. In this talk, I'll be assuming that bionanomachines are implemented based on biological cells. And this community of molecular communication have been discussing a lot of applications, molecular communication. One is computation within the body. We have some intelligent sensing systems inside the body for like a health monitoring. Or drug delivery systems, we use mobile bionanomachines to deliver drug molecules to cancer cells. Some sort of structure formation for regenerative medicine. Inside the body, they do some repairing, recovery of damaged part of the body or injured part of the body. And interfaces are also important, interconnecting molecular communication systems inside the body with outside systems. And outside system may be further connected to the internet to provide future network-based services. So we have been discussing a lot of applications. And in my talk, also in this session, we will be talking about drug delivery applications. So this is the application scenario. We have some bionanomachines, the green ones, and the target cancer cells. And we are inside the blood vessel. The goal here is to detect the target and deliver the drug molecules to the target site. And the molecular communication here is used for coordination among bionanomachines. So bionanomachines are first searching for the target inside the blood vessel, and one of them finds the target and releases the signaling molecules. It diffuses. This is this is indicating the location of the target. And nearby bionanomachine look at the signal and move closer to the source of the signal so that they can move closer to the target. And then eventually we have a lot of bionanomachines around the target where they release drug molecules. So this is the application scenario we are thinking. And for this drug delivery, we propose two molecules, two mechanisms that we can use. Here we have two types of molecules, signals. One is the repellent and attractant. For repellent, bionanomachines use repellents to distribute in the space. So in this case, there are 4 bionanomachines. They each releasing repellent molecules, and also looking at the gradient, the signal, and then move toward the low concentration of repellent. So they distribute in the space. This is one type of molecule we propose to use. The other type of molecule, the opposite, attractant. This guy finds the target and then release attractant molecules, create the gradient in the space, and the other bionanomachines move toward the higher concentration of the attractant molecule so they can get closer to the target. So we have a repellent for bionanomachine to distribute in the space, and attractant bionanomachine to come closer. Using two types of signaling molecules, we can do target detection and also tracking. This is a computer simulation. We have bionanomachines and the target, and I show you how the two types of molecules are used in target detection and tracking. So the bionanomachines, they don't know where the target is, so they first distribute repellents, and they move away from each other. So they distribute in the space. And two of them, they find the target, and they start releasing attractant molecule, so that the other bionanomachines are attracted to the target. And this target here is moving target. So it may sometimes escape from the group of bionanomachines, but bionanomachines keep using this mechanism, repellents to distribute and attractants to get together, so they can do the tracking if the target is moving. And one thing to talk about here is that the type of the molecule. I introduced two types of molecule, and for each molecule, it could be diffusive molecules or non-diffusive adhesive the molecules, which I'm going to talk here. So diffusion-based molecular communication, as I indicated in the first slide or second slide, the architecture of molecular communication, we send or release molecules that propagate in the environment. And non-diffusion-based molecular communication, the sender-release molecule, this molecule do not propagate in the environment. They stick to the surface of the environment, so they stay there. So there are two types of communication we considered here for drug delivery application: diffusion-based and non-diffusion-based molecular communication. So I think I'm repeating the same thing. So diffusion-based molecular communication, we use diffusive molecules. The molecules diffuse in the space. So it's like this. And receiver bionanomachines, they look at the gradient made by the diffusive molecules, and they move to the higher concentration or lower concentration of the molecules. In biology, this is called chemotaxis. The bacteria shows this type of feature or ability, sensing the concentration of diffusive molecule, like AHL molecule, and they move to the higher concentration. So this is one of the two. And the other type of molecular communication, in this talk I'm assuming this type of communication for drug delivery. So we use adhesive molecules. So the molecules bind to the surface of the environment, and they do not diffuse. So let's say this is sender bionanomachine, they are moving. As they move, they release the molecules. And they are not diffusive, so they remain in the environment. In a very similar way, we can create a gradient using this type of the molecule. And in biology, this is called haplotaxis as opposed to the chemotaxis. And some cells show this type of response to the gradient made on the surface. And another thing is about the functional division. The question here is whether to have the same type of bio-nanomachines for drug delivery application, or we have multiple types of bio-nanomachines communicate. And there's really some pros and cons. But in this talk, I will be focusing on this, the functional division type drug delivery. So we assume here the two types of bio-nanomachines are readers and the followers. The readers, they are responsible for making the gradient in the space. And the followers, they are carrying drug molecules. And then they look at the gradient and move closer to the target. So we divide the functionality into two types. Okay, so that is the introduction. And I will talk a little more detail of the drug delivery application. And our goal is to demonstrate the feasibility in wet lab settings, but given the currently available resources and facility, to myself, it's very difficult. So we do a little experiments and also do modeling. We do experiments. In experiments, we do some measurements. We look at the cell, how they move, and do measurements and build some a model based on the experiments. We also get some parameter values from the experiment. And then we use the model and also parameter values to in the next section, computer simulations. We want to do everything in wet lab setting, but it's difficult. So we do some experiments and some modeling based on the experiments. So the detail, a little more detail. So we first need to choose the material to use for the drug delivery. And in this project, we decided to use endothelial cells for bio-nanomachine, for a model of bio-nanomachines. Endothelial cells, they found inside our body, they are forming the blood vessel. As you see there, they are forming the internal wall of the blood vessel. The reason we chose endothelial cells, that the blood vessels are everywhere inside our body, which means that endothelial cells can migrate in our body to reach every location in our body. So they have very good ability to migrate inside the body. So we use endothelial cells as a model for bio-nanomachines. So we observe how the endothelial cells move. This is on a culture dish. This is time-lapse imaging. We take a photo every 2 minutes and see how the cell move. This is more like a random walk. It's about 14 hours, but staying almost the same location. If you look closer, it shows very interesting morphology, shape. And this is like this, and they have a nucleus, and they also have these small like fingers. These are the sensors. They're sensing the environment. They're sensing the molecules maybe on the surface or in solution. And this cell is adhesive, so they're sticking to the surface, the culture dish. They have sensors. And some following part, they extend these sensors are deciding the moving direction. Also, they have the body part, the stress fibers making the motion, and also the other side of the moving direction has some mechanism to detach from the surface. So this cell moves that direction. And just a moment. Okay. yeah. So if we put a lot of cells together, they show some sort of collective behavior. This is based on the physical interaction between cells. So they are initially packed together, and here is the open space. So they migrate toward the open space. As you see here, it's not clear, but it also shows a very interesting the morphology, the shape. Some cells are like this shape, or like here, more rounded shape, and here is more like this, and there are a lot of shapes you can find. And then if you look closer, they are changing the location. So there are some physical interaction among the cells. And in terms of the time scale and length scale, this is 100 micrometers. And initially, they are here or there, and up to here, about 800 micrometer, and they take 38 hours to move 800 micrometer. So 800 micrometer per 36 hours. So it's approximately 20 micrometer or some more per hour. And this is relatively slow compared to the, if you compare this one to the bacteria, they are moving much faster. They are moving in the time scale order second, let's say 20 or 50 micrometer per second, if I remember correctly. This one is like 20 micrometer per hour, not a second. So they are very slow. And for this type of slowly moving cells, non-diffusion based molecular communication is more efficient. That's what we thought. If we use diffusion-based molecular communication, diffusive molecules, you have to maintain the gradient for this time period of 36 hours, which means that you have to keep releasing the molecules. You have to create a lot of molecules. But if you use the non-diffusion-based molecules, you deposit the molecules on the surface and they remain there. So some molecules decay or go away, but they're remaining on the surface. So it's more effective. So in this project we use the non-diffusion based molecular communication for drug delivery. And we have done some experiments to see how the cells respond to the gradient of non-diffusive the molecule. For this one acts as an attractant. We use fibronectin. This is an extracellular matrix, the protein. There's a report that this one function as an attractant, and so we use this fibronectin molecules as an attractant. So we created the gradient on the surface. This is done by a laboratory technique, and the fibronectin conjugated with fluorescence molecules, rhodamine. It's commercially available, and we made the gradient, and we observed through the fluorescence microscopy that the gradient is formed, and then we place the cell on top of this and see how the cell moves. So these are some of the results. We have 3 cells here, and each movie, the gradient is from left to right. And these are some of the representative results. So some cells like this, they move from left to right. It looks like they're moving based on the attractant gradient. This one is not moving a lot, and that one is moving, but it looks more random than the first one. So because the cells are very probabilistic and stochastic, and a lot of noise, and also if you can look at this, the gradient we made is not that smooth. It goes up and down. So it's not clear whether this type of cell shows the apoptotic response So we have to look at some number of the cells, and we look at the averaged behavior. So these points showing, each point is showing some cells, specific cells moving direction and also moving average the direction and this distance. So here the control experiments, there is no gradient form. And here is a gradient experiment. There's a gradient, and then you look at how cells move. So in control experiments, the cells move more randomly. But on the gradient substrate experiments, the cells move more toward the higher attractant concentration. So we confirmed that this type of cell shows some half-tactic response, but it's not that strong response. So based on these results, we try to build the model of bionanomachines. And the first thing we try to do is to have some detail in the model. So as I showed you, the cell shows very interesting morphology. They change the shape, keep changing the shape. There's an existing model to reproduce this type of behavior, and we have some. Just a moment. Okay, I cannot play the movie, but this was like this. But in drug delivery application, we have a number of bionanomachines, 100, 1,000 in simulation. And this modeling is very computationally expensive. So we are trying this, but we have no results using this type of the model. So instead, we use a very simple model. We model a cell as a single point. There's no shape, no volume, and no physical interaction. We model a cell as a single point, and it's like a random walk model, but based on Langevin equation. That the point has the moving velocity. It has velocity, so it has orientation. And this is the model. So X is the location. So that one is the change in the velocity. And this one is the velocity, and the coefficient is negative. This is resistance. If you move faster, then you get a lot of resistance. And this is the noise term, Gaussian noise, Wiener process. And this is for leader bionanomachines. If you remember, the leader bionanomachines are responsible for making the gradient in the space. They move randomly to make a gradient, but they start releasing attractant molecules when they find the target. And this is the concentration, how the concentration change, attractant concentration change. So leader bionanomachine release attractant at specific rate, and also attractant concentration decay at constant rate. And for our bionanomachines, they are carrying drug molecules, and they look at the gradient and move closer to the target. And the difference between the leader and the follower, that the follower has this half-tactic term. This is like a drift to the random walk. And the model has some unknown parameter values. So we look at the experimental data and then get some estimate for the parameter values. So in experiments, we measure the location of the cell, we get the trajectory. And based on the model, we have some expression that produce this trajectory, some probability. The expression was the probability that takes this, that produces this trajectory. And we find the parameter values that maximize this probability. So for each cell from the experiments, we estimate the parameter values alpha, beta, gamma, and these are the results. And this is experimental results. This is just a copy. And if you use the parameter values estimated, we can reproduce the very similar trajectory on computer. Do I still have time? 1 minute, okay. There's another section about simulation results. So we have model and parameter values. We use the model and parameter values for our computer simulation. But I already talked most of the key parts of this work, so I will go very quickly this section. So I divided in two parts. One is for leader bionanomachines. Leader bionanomachines are responsible for making the gradient. And the second section is about follower bionomachine, carrying moving the drug molecules. So here I use the term trail network. So leader bionanomachine, they're moving and also release attractant molecules. If they release molecule after they find the target, there's a bunch of leader bionanomachines. They create the network like this. It's like the ant, group of ants depositing the pheromones. So we here use the term trail network. And we look at how the trail network are formed. We look at the dynamics. And this direction indicating how the follower bionanomachine move in the space. In some cases, the trail network forms some local optimum. It's high, so the arrows are pointing each other, which means that the follower bionanomachine, if they are introduced, they are stuck there. They fail to move to the target location. Also, if we change parameter values and some model a little bit, then we can create a different dynamics and different distinct network like these or that one. So now we have gradient made by the leader bionanomachines. Now we introduce follower bionanomachines. We put together leader bionanomachines randomly moving and making the trail network and follower bionanomachines carrying drug molecules. Eventually they're closer to the target. Okay, so through the computer simulation, we confirmed the feasibility of this model. Okay, so there are some other detail, but I will skip. So I presented application for drug delivery, and there's some future work. Okay, thank you very much.
Well, you all agree with me. That's been an outstanding presentation. We know where the field is moving forward now. Let's get some very quick and focused questions, please. The microphone.
I wanted to ask you, since you're the first cell which is looking for the injured cell or whatever it is, knows when it has met the target, right? Is it possible just to think at the only just one type of just by switching that kind of pheromone which has to be released at the time it has followed the target? Is this feasible?
Yes. Actually, there's another work actually before this one in which we proposed one type of bionanomachine doing everything, making gradient and also carrying drug molecules. Yes, possible. Yeah, it's possible. Yeah.
Yes, here, please. We have now the microphone, so I have to switch the microphone then.
Thank you, Andrani. Thank you, Tadashi. Very interesting, very short question about scalability of your model-based simulation. Scalability in terms of you're assuming that the target, it's punctual, it's a point in space. How the simulation would perform if the target would be relatively larger than the nanomachines and irregular in shape? Can you comment on that?
Actually, target is not a point in space. It has some
volume.
Yeah, volume. But the bionano-machines in this case are point. Machines are point, bionano-machines.
But still, the size of the target is relatively, well,
In the simulation
machines are
setting, we
punctual.
assume the 1 millimeter for the target size.
Okay, thank you.
Okay. Yeah, very quick and very focused question, please. Okay, here.
Can I just clarify something? My understanding, and this might be wrong, is that when you trace the movement of these nanomachines, this trace pattern is inferred from image processing, right? It's not real, right? It's traced from the gradients, and they do some kind of machine learning inference, right? No?
It's from a photo.
But it's a sequence of photos,
There's no
right?
Inference.
Okay. Oh, it's only because you have one, but if you have multiple, then you might need to do inference, maybe.
We can still get the photo.
Okay.
Get the group of cells.
Okay,
We
okay,
can see where the cell is.
okay, all right.
There is no machine learning.
Okay, apologies.
Okay. Well, thank you very much. Well, please join me to thank the speaker once again. Our next speaker is Professor Kostas Pitris, Professor of Biomedical Engineering at the University of Cyprus and the GEO Center of Excellence. And he is going to present optical and multimodal interfaces for molecular communication systems.
Well, thank you for inviting me here. Actually, my work is in optical imaging, optical diagnostics, and with Andriani, we do a lot of molecular imaging, which in a sense is actually one building block in what this community calls molecular communications. So I will attempt in a very short time to actually present an overview of various optical techniques that have been developed over past 3 decades or so, which could be useful in molecular communications. The presentation is by no means complete or very detailed because of the time limits. But if you want more information, you can interrupt me or you can find me later and I can give you more details that your stomach can handle. So the idea is to actually use optics or the optical signals as carriers. And it has been proposed that optics or optical signals can play a twofold role. One is interfacing between the micro, nano, or molecular machines, devices, and also enable interconnections between individual nodes. I will be focusing mostly on the interfacing in this talk, and I will talk about light transduction, which is essentially the conversion of one signal to another. That's the definition of transduction. And in our case, is the conversion of a molecular signal into an optical signal, or the reverse, an optical signal into a molecular signal. So why optical? Well, light offers many advantages. You can do imaging or detection or sensing with very good resolution. It's very sensitive. It's non-ionizing if you're planning to go in vivo, which ultimately should be the goal of most of the applications. You can multiplex light because you can use different wavelengths to detect different modes of sensing simultaneously. You can do real time. It's inexpensive, so it's not like setting up an MRI or a PET. And you can incorporate it easily into micro or even nanoscale devices. It has been very well studied for biological systems, so the adaptation to new applications is actually not very complicated. I'm not going to say easy, because nothing is easy, but it's not that complicated. One of the major disadvantages of light is that it cannot penetrate biological tissues or material very deep. But that, to a large extent, can be mediated by using delivery devices, such as fiber optics. So you can easily integrate the delivery of light and the collection of light into an endoscope, for example, and examine the internal cavities of a body. Another interesting advantage or useful advantage of optics is that you can, due to the sensitivity and speed of detection, you can do real-time imaging, which allows you to perform investigations in space and time. So you can do, there's a lot of interest now to do spatial temporal mapping of your molecules, your sensors, and that gives you a better understanding of not just the local effect, but actually the interactions in space. So where could light become useful? Well, you can use it to interface and monitor all the steps of a communications process. So you can monitor the encoding and the decoding process. For example, gene activation. You can see gene activation. You can target the genes or molecules that actually participate in the encoding or decoding, the same way you can target the molecules that actually participate in the reception or transmission of your signal. And you can also monitor the dynamics of this transmission, and I will show some examples of techniques. So gene activation, there's a lot of work done in this area. You can transfect your encoder or decoder gene with a molecule that can be detectable by light, and there are two common techniques that are used. One is bioluminescence, where a molecule called luciferase enzymes are encoded, are transfected to be co-expressed with your gene. And what luciferase does is that when it interacts with its substrate, which is luciferin, it can create light. So you have a model, you have a molecule that actually emits light by itself. This is all based on the molecules derived from fireflies, so the insects that actually emit light. This is a very useful model, but one of its disadvantages is that there's a very slow decay. So you can see the emission of the light, but it's not going to clear within seconds or milliseconds or seconds, but within minutes. So it's good for detection of the end result, but it's not particularly well suited for communications in the sense of fast communications, or at least as fast as molecular communications can go. Another option is to actually transfect the cells with a fluorescent protein. So a fluorescent protein is co-expressed with the gene, and in that case you have another means of detecting the expression of the gene, except now it's required that you have a laser to excite fluorescence from the fluorescent protein. Again, a very useful application, a very useful tool. The decay of fluorescence is fast. However, once the fluorescent protein has been produced and it accumulates in the cell, the clearance of the fluorescence protein is slow. So again, here you have an issue of how fast of a communication model you can build based on these gene activation methods. Another option or another application of optics is that you can use optical sensing to see the genes, the molecules, or the structures that participate in the communication. And the first step is to actually label those structures or proteins. And in order to label them, you have to actually find them. So step number one is targeting the protein or enzyme or DNA of interest. And targeting can be done in various ways. One method is to use antibodies, which are created to actually attach to certain structures. You can use small peptides, which actually go and attach to receptors. Or you can use aptamers, which are small sections of DNA or RNA, which attach to DNA or RNA, and when attached, actually you can emit fluorescence, and that way you can detect the presence of those macromolecules. Or you can have your own, the molecule itself that is doing the that's participating in the molecular communication be activatable, and in the presence of enzymes actually change its structure or cleave parts of its structure in order to fluoresce versus not fluorescing in the absence of those enzymes. So there are all kinds of techniques that you can use to target either receptors, DNA, enzymes, or even structures in the cell. Now, once you target your molecule of interest, then you can label it optically and use optics to detect it. And the most common technique is actually to use fluorescence. So you can make you can attach fluorescent moieties to many peptides. You can detect those in a fluorescent microscope, in vivo, or in any other way. This is a very inexpensive and biocompatible way of detecting things with a fast decay time. Fluorescence decays fast. However, fluorescence has a disadvantage, which is photobleaching, which means that if you illuminate it for a long time, it will not fluoresce anymore. However, that is not necessarily just a disadvantage, because in a few slides from now, I will show you a technique that is based on photobleaching, and it's actually very useful. So, but depending on what you're trying to do, photobleaching may be a problem. Lately, there is a lot of work on metallic nanoparticles, usually made of gold. The choice of gold is because they're non-toxic, and they scatter light very effectively. The disadvantage is mainly in the in vivo distribution of the nanoparticles. They can be fairly large, and also the toxicology of nanoparticles not made of gold. There are cases where silver or copper may be more useful optically, but those metals actually have toxicity issues, and even gold nanoparticles, depending on the size, can cause toxicity. Lately, or more recently, semiconductor nanocrystals, so-called quantum dots, have emerged as a very promising optical label. They are very efficient, very small size, and they can be tuned to different wavelengths, so you can multiplex. I don't know if it's shown here, but different colors in the same animal. But they have a very significant disadvantage, which is a problem in vivo, in that they are toxic. So the quantum dots traditionally made with cadmium telluride are actually toxic, but there's a lot of research in replacing those elements with rare earths, which may provide quantum dots with less toxicity. Fluorescence, as I said, can also be a useful tool, or the effect of photobleaching in fluorescence can be a useful tool in detecting dynamics of the dynamics of molecular communications. So the fact that you can actually turn off the fluorescence of your molecules can be useful in what sense? You have cells, for example, that communicate, perhaps through gap junctions, and you have this green fluorescence that you measure with your microscope. You can go in with a high-intensity source and actually photobleach one or more cells. This one here. So for example, that cell was photobleached, and intense light was used to turn off the fluorescence in that cell. Then you can actually watch and measure how the fluorescence recovers. So how the signals that are coming in actually reactivate, either produce new fluorescent agents or fluorescent agents flow in through gap junctions. And you can use this recovery of the fluorescence to understand the dynamics of the flow. This is called fluorescence recovery after photobleaching. And it's very useful for cell-to-cell communications. Another technique that has been used already in molecular communications is a technique called forced resonance energy transfer. Sometimes you can see this as fluorescence resonance energy transfer, so the F changes, which is a useful technique, again, to monitor dynamics. And it's based on the fact that you can make you can attach fluorescence moieties to two proteins, protein X and protein Y, but you can make the fluorescence portion of protein X be a so-called donor to the acceptor fluorescent protein of protein Y. So what that means is that the acceptor molecule can absorb the light emitted from the donor. So when you have just the protein X by itself and you excite at the right excitation wavelength, you'll see an emission. For example, in the case of the figure, I say the emission is, you excite with 395 nanometers and the emission is probably towards 400, 450. So you can detect that and you can measure it. But if there's protein Y in the same mixture and the two interact and they come close together, so they attach to each other, the light emitted from the donor will be absorbed by the acceptor, and the acceptor will fluoresce, but now at a longer wavelength. So it will absorb the 400 nanometer light and emit around 500. So you can measure the ratio of donor and acceptor, and you can measure the interaction of donor and acceptor. So if there is interaction or not and how much. And FRET was actually demonstrated as a communication channel. And me being an electrical engineer, I couldn't resist putting the eye diagrams on the slide here, but those were actually the eye diagrams from a communication channel based on FRET, molecules interacting in the resonance energy transfer mode. You can also use light to build switches. So here's a little bit of the opposite. So now we're talking about using light to change, is a transduction of light to molecular information. So we're using light to change the molecular makeup of the target. And these switches can be made so that, first of all, you can use any type of trigger. It doesn't have to be light, but you can use triggers such as calcium, of the well-known calcium switch that is has been described in the literature, but it can be pH, temperature, or even light itself. And yet you can switch molecules from one state to the other. You can switch molecules from on, off, for example, or from transmitting in one wavelength to transmitting in another wavelength. And one group has even shown that you can do logic with switching lights. And of course, in their example, they talk about cell 1 and cell 2. They mean a cuvette. But there is no fundamental reason why cell 1 and cell 2 couldn't be cells, biological cells. So as I mentioned, the primary limitation of light is actually the relative limited depth of penetration. So over the years, different groups have developed solutions around this. One solution is to create the delivery optics that can take the light where you want it. Another solution is to create contrast agents that are multimodal. So in this case, you can take advantage of modalities like CT, MRI, or SPECT, PET, to perform a survey of large areas, even the entire body, but then go and home in with the optics to get the sensitivity and resolution that you would want in trying to find, say, the very early stages of cancer. And there are several combinations of multimodal agents, optical and MRI, or optical and PET, or optical and CT. I just have some examples here just to show that it's indeed feasible to combine either paramagnetic moieties with optics or radionuclides with optics, and then you can use the same agent using two different modalities. Now, another important contribution of optics is that you can use optical techniques as your interface to the outside world. So you have all these exciting processes going on at the molecular level, but somehow we have to monitor what is going on, and we have to know and have a way to interact with these molecular networks. One way you can do it is by optical sensing and optical imaging. The most common technique used is microscopy, but there have been other techniques developed that can be useful. I will present just some of them as a very brief outline here. First of all, fluorescence microscopy has been the workhorse of discovery in biology for decades. It's simple and well understood. There are limitations in microscopy when it comes to thick tissue sections, so other techniques have been developed to allow sectioning, such as confocal microscopy, where you scan a pinhole over your tissue to reject out of focus light, or multiphoton microscopy, where you use very short laser pulses to concentrate the excitation only at the focal plane, since it requires 2 photons to absorb and create fluorescence. And those can provide sectioning of thick tissues, and you can see the cells individually. And I couldn't but add this slide for 2 reasons, because these guys won a Nobel Prize, but also it's the only technique that allows you to go below the fundamental resolution limits of optics of your lenses and image items that are, or targets that are in the nanometer scale. It's fluorescence essentially, but what they do is they excite fluorescence just like they would normally excite fluorescence, but then they go and deplete the excited molecules by using a donut-shaped beam, which leaves excited molecules only at the very center of this donut, which is a nanometer scale area, and they measure fluorescence only from that nanometer scale area. And the results are actually impressive. I mean, you can see confocal, and This is not what we're used to seeing in confocal, but this is because it's expanded to a larger scale. And then you can see the individual molecules of the, I think these are in pore complexes. Yes, these are pore complexes. So it's individual molecules. More on the macro scale, you can use fluorescence to do 3D imaging. And this is done by so-called fluorescence molecular tomography, where fluorescence is measured from around the sample, and then a reconstruction algorithm is used to create a three-dimensional section, and that is overlaid actually with CT, but you see the fluorescence is here. You can measure absorption of molecules, and you can measure the absorption by actually viewing the sound waves created after the absorption of light, and that's called photoacoustic tomography. And you can do it in 3D, so you can measure absorption of different molecules, and you can do it simultaneously with multiple wavelengths, so hence the extension to multispectral optoacoustic tomography. And if you're looking to correlate this with microstructure, there's a technique close to my heart that is called optical coherence tomography, which is the analogous of ultrasound but with a laser light, where you can get fine detail at the limited penetration that is allowed by light. And also, by looking at the spectroscopic content of this light, because it's a broadband light, you can actually get some information on the molecular constituents of the tissue. So in closing, light is a good communication medium. I hope you are convinced. There are a lot of challenges on how to adapt this to the molecular level detail that is required here. But there are also many new and exciting materials and structures that can be used, like carbon nanotubes, graphene, molecular LEDs, etc. But I couldn't help adding as a major challenge here, actually forming and truly integrating the multidisciplinary teams that are necessary for taking this area forward, and especially into the clinical applications of molecular communications. Thank you very much.
Thank you. Let it be the light. Okay, that was the first question. Okay, take 1 or 2 very focused questions. Yeah, Professor Lee over there.
Thank you. Thank you, Kostas. If you are using, say, photo switchers, several, and you may also get some information about the sort of biological time of a process that could be different from the time that you can measure externally. So it can be for a cancer, for instance, or for some other process. What is your thought?
Yes, one of the challenges that we've been discussing a lot yesterday actually is the sensitivity issue, and if you have only very few molecules, how do you detect them? Switches are actually a very interesting way of seeing these changes and actually accumulating the changes so that are detectable, so that's one thing. You can also do real-time imaging if you have enough of a signal, and you can, and especially since you can multiplex these, you can have a switchable, so your events can be steps, switchable steps in wavelength, for example, which you can monitor in time. So you can think of, I mean, your imagination is the limit here. If you can engineer your system any way you like it, you can put all these switches in it, you can have wonderful applications.
Okay, back.
Yeah. Okay. No, I don't need that. Oh, okay.
Please.
Okay, sorry. Yeah, sure. One question and one comment. Question is, have you encountered, when you do your, research in this field, have you encountered anything for molecular to quantum entanglement channel transduction? So this is the question. And the comment is about the fretwork. I'm happy that you liked it. And actually, we had passed Bob Dylan's House of the Rising Sun song over the protein molecules, and we submitted that paper, but Nature said that, no, you cannot do that. You have to get the copyright and all. So we had to change the song, but you can download the protein past Bob Dylan song over the web if you like.
Okay. So what was the transmission rate to get the song through?
176.4 in that experiment. Kilobits per second.
Okay. Just curious what it takes to transmit a song.
Yeah.
Quantum entanglement, we haven't. Usually our work so far has been molecular imaging for cancer diagnostics, so we are more on the micro scale. But it will definitely be one of the challenges if you go down to the pores and receptor structures.
Okay.
Thank you. Again, very nice presentation. I totally agree with some of the challenges you mentioned, which is we need to reduce the hardware size. This also points to Pietro's comment. I think that one of the challenges is, sure, with light we can see many things. Sometimes we see too much. You illuminate a tissue, you see responses over tons of cells. Can you comment a little bit on, how small can you get with, let's say, nano devices for light and actually try to overcome some of these problems? I mean, are you familiar? I mean, you mentioned some works on nanoparticles. Can you say how many cells can you target at once?
Okay, well, there's two ways you can improve your resolution, let's say. One is to actually make your light beam as small as possible. And that is limited. I mean, there's only so much you can focus a light beam. Even if you play tricks like the stimulated emission microscopy, there's still so far down you can go. The other way you can improve your resolution is actually by targeting single molecules. So if only single molecules are actually labeled, then it doesn't matter if your light beam is big. You're still measuring a single molecule. And there's a lot of work on single molecule and single nanoparticle detection in the literature. It makes the targeting harder because you have to assure that you're targeting single molecules and you have to assure they're not close enough to cause clumping, and then your resolution is worse. But theoretically, you could go down to single molecule. Now, we say that, but as you go to in vivo cell live systems, it becomes increasingly difficult.
Thank you, Andriani. A laser focus question, very short. Thank you, Kostas. You stressed the potential of these techniques to downscale to the microscale. Could you please comment on the energy requirements of this, in terms of the energy requirements of the laser source, the propagation laws of light, the lack of efficiency of the devices?
Well, the efficiency of the process is always an issue, so fluorescence is not 100% efficient, obviously. But it's still, I think, efficient enough to get good sensitivity. The question is how much do you want to shrink your device? Why you want to shrink it? Because you may not have to shrink it as much as we think. Maybe targeting is a more practical way of achieving better resolution. But yes, in some cases, you may want to have implantable devices, for example. And actually, we had a very interesting discussion earlier that there are 10 micrometer implantable lasers now.
But is that feasible energy-wise?
Energy-wise, you have to somehow have an external source. Mm-hmm. It's actually feasible. There are many ways you can transfer enough power. I mean, they do it for cochlear implants, which require, I think, a couple of orders of magnitude more energy than the 10 micrometer laser.
And during the coffee break, we can talk about your thoughts about the potential of the terahertz band, no man's land. Right? Thank you.
Okay, before inviting the next speaker, I just want to point out at Kostas' last sentence. Form and truly integrate the multidisciplinary teams necessary for breakthrough advances. So we have the time to discuss tomorrow. Well, unfortunately, the next scheduled speaker cannot be with us today for health problems. Dr. Alan O'Riordan from Tyndall Institute here in Ireland. We all wish him a speedy recovery. And I'm inviting to the floor Professor Ilanko Palashikam, if I pronounce well. I just know him by Ilanko, okay. And he's going to talk on communication theoretical approaches to model neuronal networks and predict function. Thank you.
Okay, so this is going to be slightly different. Again, looking this problem from communication theory, information theory, and see whether how we could contribute to make some, first of all, to understand the behavior of neuronal networks, and whether we could use this to some applications like how to stimulate neurons from a distance. So basically, two things I would like to touch upon here is to see how much information is possible to transmit between two neurons. This would be, sounds very theoretical work, but hopefully that would be useful if you want to sort of address certain things between neurons, and then also how you could stimulate that. So I put up on this slide basically to try to see, we get a lot of external stimuli, and then our brain is able to process this information, and hopefully that could be also information processing and understanding actually leads to some kind of activities or creative thinking. Everything is sort of related to that. So I'm a little bit interested to see, for example, the last one here. Is it possible to use RF or communication theory techniques to understand the brain communication networks? So, sorry, before I go there, I just wanted to also show, I don't know whether you are able to see this picture here. It's basically a small pacemaker device, and there is some wires going up all the way to the brain. It's basically used for Parkinson type of stimulation. So you're basically using electrode to stimulate certain regions in the brain for Parkinson application. So I don't want to explain, maybe a few words about brain and neurons and where we are. Basically, we are a little bit interested about hippocampus part. Especially, I will talk a little bit about the Alzheimer problem and which part of the brain is associated to that. And that's the reason that I'm touching upon here. And in addition to neurons, we have a lot of other things. It's called glia cells, astrocytes, and then we are also using astrocytes as part of the communication. So let me come back to that a little bit later on. When it comes to communication systems, biologists actually, or neuroscientists or neurobiologists, they have kind of worked out this. They explain how this works, and there was a Nobel Prize for that in 2013 for 3 scientists. Interesting part actually is that they use slightly different terms than communication people like me use. So we were trying to understand how this kind of works. It says about that there is something called vesicle traffic. There is something called fusing these vesicles with cargo molecules, and then how the cargo molecules going to be delivered with the precision, so that there is a communication aspect taking place here. And we are trying to see whether we could explain this using a classical way of looking this from a communication theory point of view. So we are trying to sort of map some of the terms coming from biology to our field and see whether we could use our terminology to understand this communication aspect. So we will talk a little bit about transmitter, for example. We'll talk a little bit about channel. I'm not sure that I'm able to point it here. No, it doesn't look like that. Okay. So there's something called channel, there's something called receiver, something called front end, and so forth. So don't think too much about that, but it's basically trying to sort of map certain things so that we could use the tools that we already have in communication theory and information theory. So what is actually, and also in biology, people, of course, with engineers, they are also trying to use circuit models to explain these things. Circuit models comes from Hodgkin-Hartlick's models and then has been extended to other type of models. What is actually missing that we have seen, especially coming from communication theory point of view, is that normally we are more, randomness, some kind of stochasticness is missing. Typically, all these things are differential equations and deterministic. And I understand that why they are interested in deterministic, because they do some measurements and they try to do some fitting, and they are not so much interested about the noise and all these things, but what is the steady state information and how this could be modeled into a circuit diagram. So we are a little bit interested how we could come up with some kind of stochastic way of looking at this and some dynamics, so that this could be useful, for example, to excite certain things or to convey certain messages, maybe using voltages or concentration levels or whatever that you want to have in your activation scheme in the sub-threshold way. And that's basically the idea here. So there have been some works coming from our lab, and also other colleagues have actually also have trying to look into this using a slightly different way of approaching the problem, and including some kind of probabilistic model that could sort of capture this dynamics. Let me not go into all the details, but I think people understand that this looks more like a circuit diagram with some kind of amplification and with some noise coming here, and then it will be transmitted, maybe not amplify and forward with the noise. Hopefully the noise is going to be removed. At least the system is able to do that. So again, looking this from communication engineering point of view, we use these terms like transmitter, channel, and receiver, and also the noise, that we need to understand how the noise is going to be associated to this. So that's pretty much interesting to look into that problem. So far, I have shown you only single neuron with a channel to the next one. But in addition to that, you also have, what I mentioned to you, that astrocytes. And actually, quite recently, or maybe a few years ago, seems to be playing a huge role when it comes to the communication and regulation of the system. And we understood that we need to consider that. It is not only looking at single neuron to another neuron, but also how the astrocyte is going to play a crucial part when it comes to the communication. That seems to be quite important. So that's the reason that we have also taken to that. So in this case, for example, it could be a calcium wave that would be propagating through the channel. In addition to that, we would like to see whether we could excite the neurons using some kind of a small device like a nanomachine. It could be a biological machine, it could be based on a nanomaterial type of device, or an external field that we could apply, so that this would be picking up that information and subsequently driving the neuron. Yeah, so this is actually the indirect way of stimulating through the astrocytes, and how this actually is taking place. I don't want to go into all the details. You can read maybe from the different papers I'm listing under here. And important part actually is that this create what they call it as a tripartite synapse. And this actually is quite important to understand how these different communications, you have both chemical and electrical signals coming in, and how these are going to have feed-forward and feedback loops in a more stable way, so that you have the communication. So communication is taking place both using chemical signals and also electrical signals. So understanding different components that you already have, and then we were trying to understand how this actually works. Both the intra-neural part, so that's within the neuron, for example, and then also between neurons. So both you have the electrical and the chemical signals coming in, and how this diffusion and everything is going to take place. And importantly also how your receivers are going to be. There are different type of receptors are there, and how this kind of multiplexing is taking place, so that when you have a voltage inside and then you have a voltage outside, or receiving voltage here, then you're trying to sort of map this into information rates. This sounds very theoretical. I apologize for that. We are not able to sort of verify that in experiments yet, but we have shown that, at least using information theory, that synaptic communication, so including the tripartite neuron systems, so between two neurons, including the astrocytes, that we could have a maximum information rate, so maximum amount of information that you could transmit per bit is around 8 to 9 bits per second you could transmit. So some information you can do that, of course. When you have many, of course, then it will be possible to transmit maybe high data rate information if that is needed. I'm not sure. So that may be the case. So we looked into this, so we understand a little bit about how much information we could transmit between two neurons. So I'm a little bit interested about how I could sort of not only transmitting information, but I could use maybe some way of stimulating neurons maybe for some applications. So information coming from one neuron or one device to a neuron, for example, it could instruct what it should do, for example, and then trying to see how I could also stimulate it. So there are different ways of doing it. So when you have an invasive way, for example, an intact brain or something like that, you would like to inject with electrical currents using electrodes, for example, as I showed you with this, the pacemaker for brain, for example. Or you could also use flashing light, so it's called optogenetics. I will come back to that with one slide on that. Or I want to do something from outside without putting anything inside, like an invasive way. Maybe I would like to inject a molecule or something like that, but not like in a surgery that you need to put an electrode. So that brings us to look into maybe using electromagnetic signals, whether it's possible, and whether we can find some examples on that. So basic idea is, can a biological system work like a demodulator? So you have a transmitter, and can your brain, for example, receive radio signals or electromagnetic signals, and it's able to demodulate and maybe understand the information. Whether it's possible, we don't know yet. And also whether you could do this without having any thermal effects. So that's another question that we would like to answer, that you really can go through the blood-brain barrier. And ELF stands for extremely low frequencies. So when you transmit, for example, in this room, we are having Wi-Fi routers and everything. So you have a carrier frequency, much higher frequency. Maybe typically it's in gigahertz here, or in megahertz. And that's basically to do with the antennas that we can design. And the information rate encoded into this modulation frequencies are typically much lower. So it will be something like in tens of hertz or maybe hundreds of hertz, depending on voice, for example, on audio information or whatever. So these are much lower frequencies. So we are thinking about whether we could use a carrier frequency to transmit some type of instruction, but the instruction is of course in the low frequencies. and they might be able to interact with the neurons. So this is another example. This is not our work. It's done by another group. Of course, they published in Nature. So this is actually is using the neuromodulation, using opto, and perhaps also using some kind of RF type of techniques there. I will not go into that. But I was a little bit inspired by this experiment done by a group in Florida, in the US, where they want to study mouse excited by a normal mobile antenna and see what other changes is taking place in the brain. And basically they were thinking that this is going to sort of cause cancer maybe. But interesting part actually is they had two types of mice. One was a normal one and another one was a transgenic Alzheimer. So they inserted this Alzheimer's disease on this mouse and they did this experiment. So they put an antenna here and radiated this mouse for different times per day, and they did this experiment over a very long period of time, but exposure rate was much lower, typically like what you are using in a mobile phone. So when it comes to Alzheimer, there are a lot of discussion going on. It's not very well understood yet why this is actually taking place. And we understand that there is a disruption of communication. So this is actually the healthy cells there, and this is the Alzheimer type of cells where you have degeneration. And the degeneration is due to something. There are several hypotheses here. And something people are strongly believing that these beta amyloid deposits or plugs are basically disrupting these neural cell structures and also communication channels. So basically, they're depositing on this axon. So these are the axons. And after some time, they disintegrate and they don't get enough protein so that they are unable to grow or sustain, and they sort of eventually become something like here. So when they explain their experiment, we asked them, why this is happening, what do you think that it's able to disintegrate? Is it possible to come up with some kind of scientific explanation? And that's the reason that we got started with this work. And then we, as engineers, we try to sort of understand whether it's possible to sort of shine electromagnetic signal to the brain. Of course, we know that's been done extensively when it comes to mobile phones. The mobile phones radiation to the brain and everything. And our interest to see whether we could apply some kind of EM wave. And subsequently, the EM wave should induce a voltage, and this voltage should sort of induce a current, and subsequently you will have a neural activity. So whether it's possible, whether there is a scientific theory behind this. So that was the reason that we want to start with this work. And then we try to understand, using a computational model, what type of frequency might be useful for that. So we found out, for example, the frequencies in 300 to 400 megahertz might be useful. Maybe also in 600 to 700, and maybe also in 800 to 900 megahertz also can be useful. So mobile phone CDMA type of frequencies are around this frequency here, around 800 to 900 megahertz. So that's actually showing that how much E field that you could induce, and subsequently using the theory that we can estimate how much current we can induce inside from outside field. I think I'm running a little bit out of the time. Yeah, sorry about that. So basically, I'm skipping a lot of information here, just explaining maybe the conceptual part here. So from that point of view, we designed something called a synthetic nanomachine. So this could be a synthetic cell, or it could be an artificial type of device that could have interaction with the neuron. So neuron is this size here, and then you could have interaction using the gap junction, and then you could induce, for example, certain number of currents, and then using our stochastic model with the sub-threshold type of approach, we could show that after some time, so if you apply a high enough voltage, of course you will have a firing. But if you want to apply low amount of voltage, just because of these are going to, hopefully this could last maybe 10 years. And we would like to apply them slightly differently. So we are not applying them in a simultaneously at the same time instances, but different time instances. And it seems that there is some kind of memory or some kind of activity taking place, but it takes some time. So given those time and everything, you might be able to get an activity as a firing even when you are within the threshold level, the sub-threshold level. So that's what we try to show, that sub-threshold is interesting, using different timing is interesting, some kind of stochasticness we should use so that we could even drive a neuron. I think I will skip this part. It's basically, just to summarize this, that you have different techniques. So you have non-invasive techniques, you have invasive techniques, and how this whole thing actually can be put into something like a block diagram so that people like us can understand. And basically, maybe engineers like me, coming from this type of background, we are working in a very low layer of this structure. Of course, you can say that biology people understand even much lower layer than this, but then there are some techniques and theories that we could use, how this the transmission between neurons, the network part, I didn't touch upon that here, and hopefully what would be the effects, how we can understand this actually has this effect. And then of course you need to collaborate with some other people, maybe clinicians or psychologists or something like that, if that will be useful. Thank you.
Thank you very much for respecting the time. Oh,
Yeah.
right.
Thank you.
Karisto.
That's in Japanese, so
Yeah.
I think you understand that.
Yeah, okay, very lively. Okay, can take focus questions.
Sorry.
Here, people who have, yes?
Very nice talk, thank you. So you said you're doing stochastic simulations. Could you say a little bit more about the nature of that noise and precisely where you add it in the system?
The noise part, yeah. So the noise actually comes from the molecular noise. So you have the synaptic shot noise and molecular noise. So these are basically noise that we believe that it is not like an additive noise that we have in Gaussian binary channels in wireless communication. So the noise basically comes from the molecular parts. So that's the basic idea, that we would like to include that noise when we do the simulations and the calculations. The whole model is actually taking into care of that.
The number of molecules, you mean then?
Sorry?
You mean the number of molecules?
Number of molecules, yes. Yeah.
So, just a quick. Just a quick thing. The current model is that all the spikes are coming from the somas, and then the dendrites are passive elements to the other. There was a paper in Science in March 2017 from a group in UCLA showing that actually only 10% of the spikes are starting from the somas, and the majority are from the dendrites that are active elements and not anymore passive elements. Is this going to challenge all our models, current models of neuronal circuits?
It's a very, first of all, thanks for pointing out that paper. Actually, I didn't see that work. I haven't read it. So I think it will be very interesting, because if you remember, if you go back maybe 6-7 years ago, people thought, the neuronal synaptic part was basically doing everything, and the glia cells were not sort of, involved in this process. And then people start looking at that, and the abundant number of cells were there, and then they're trying to sort of, go back and do the modeling again. And maybe this new, insight coming from this work, hopefully will change a little bit about the way that we are doing the modeling again. And I haven't, to be honest, I haven't seen that paper, so I don't know about it. But it's very, Thank you so much, Pietro. Yes.
Yeah, that's the last one.
Sorry, come back to the noise again. Could you also identify if the molecular noise you're studying affects the rate of the firing in a much, for example, if the rate of the firings are very low frequency, so to speak, or very high frequency?
How the noise affects that part?
Yes.
The noise definitely affects the rate, amplitude values, but I cannot say that how this affects, because the noise seems to be dependent on the input signal. So typically what we understand from communication is that the signal that we are transmitting actually does not depend on the channel noise. Here the channel noise is dependent. So it's a correlated noise. So I cannot sort of divide them in the traditional way that we have been working maybe in communication theory. So that's something that how this affects is dependent on that. That's the reason that they call it a shot noise, I think. So I cannot answer that. Beyond that, I understand that it affects, but I don't know exactly how much it will affect just because it's correlated with the input signal.
Okay.
Yeah.
Okay.
Well, thank you very much.
Thank you. Thank you. Okay.
Well, until the slides are on, I have to say that I have only 2 minutes, for my presentation. I have to pay the toll for, a very stimulating and exciting discussions by all the speakers here. All right. Oops. What's going on? All right. Some 150 years ago, a French physiologist, the renowned father of modern physiology and experimental medicine, Claude Bernard, defined medicine as a fragile art applied on solid sciences. I cannot think of any better example as of what's going on here today. He was the first to define the milia interior, the interstitial, the microenvironment, so I think he has contributed a lot already to molecular communications. Well, since the time Isaac Asimov, defined nanobiotechnology through his fantastic voyage where he described a nano-subject submarine with 4 men, 1 woman, going through the circulatory system to find a clot and destroy it. Actually, that was the first application of nanobiotechnology and perhaps nano networks, if you just think of it. We have gone a long journey, a fantastic voyage. So we're at the point moving from nanomaterials and nano devices to nano networks and nano systems. And proud enough, we can speak of the realization of the great promise, go back, to Richard Feynman's talk. And Nobel Prize in Chemistry last year was given for the design and synthesis of molecular machines. And I say, yes, we are going to the lower limits, and we're at the point of molecular nanomachines and nano networks at the fore. So the title of my talk was, systems for validation and translation. What do we want to validate and what do we want to translate? The first is to validate molecular communications theoretical models. I'm sure that's what all of you want to find. So we need basic testbeds enabling quantification of model parameters. So what do we want to measure? We need to define that. And how do we define and analyze molecular signal metrics in biology? And then it's translation research, which has to do with quick transfer of data from the lab to the clinic. So it has to do with disease diagnostics and therapeutics. In terms of diagnostics, we have biomarker detection and quantification. So we need to have models for spatial and temporal disease evolution and define, design, and synthesize the suitable bio-nanomachines and nanonetworks. We're coming to to therapeutics, tumor pharmacology. We need drug development testing models adjusted to molecular communication era, and need to recapitulate the in vivo conditions, applying principles and methods of molecular communications. Therefore, the challenge is how to translate, I would say, layered architectures to multi-layered tumor constructs. So I don't know if any of you is familiar with the so-called roadmap to novel drug discovery and development, but you start from the basic discovery to preclinical, clinical models all the way to translation. So here, I translate this to the roadmap towards biomimetic tissue engineering and tumor modeling systems for drug development. So we try to build bridges across natural sciences, engineering, and healthcare. So starting from material design, fabrication, cell expansion, all the way through clinical development. And here we have a great role for computational omics. With genomics, proteomics, metabolomics, and of course economics. We need money to do that. Systems biology. And we have new emerging disciplines, which are the multifunctional growth factor nanoparticulate systems in terms of combinatorial signaling in vivo and polymer libraries. And we see, different other areas coming up, like material structure modeling, computational fluidodynamics, and developmental molecular biology. And we need, of course, in vitro validation and in vivo validation. So three-dimensional tumor models is emerging of tissue engineering and cancer biology in order to have robust interrogation of drug delivery. So the challenge here is to reconcile the time scale of few days and weeks we can have in a typical in vitro study with the dynamics of clinical cancers, which take many, many years to grow. That's a big problem. And the major technological issue here is we need the proper cells to remodel their environment, changing metabolite gradients, mechanical forces, and finally disrupt a predefined microenvironment. So what we have here, it's a very standard way to develop three-dimensional tissue constructs and tumor constructs based on nanofibers, electrospin nanofibers. Electrospinning is a very powerful technique where we use an electrical field to direct polymers, in very fine, structure and make some complexes mimicking the interstitial tissue and extracellular matrix of the cell. So this is a technique we use in the lab. So we can have layers of nanofibers at the appropriate cell, another layer of nanofiber, and have that structure here. And if we go to translate into tumor, take a tumor biopsy, isolate the cells in culture, and go back and restructure the tumor. And if we have technical issues, go back, test with normal cells, answer those questions, and go back. Coming to an actual disease model here, in order to see how we can develop models for a disease where molecular communication theories can be applied and give directions and solutions in diagnosis and therapy. Glioblastoma multiforme is the most malignant human tumor. It's a brain tumor with, high heterogeneity, with very poor, outcome for the median survival. So there is much to do over there. And serve as a prototype for the study of tumor initiation and recurrence. And the first thing to take into consideration is the powerful glioma stem cell. It's the first cell where cancer arises from, and it has the potential of cell renewal and progression and differentiation to different types of tumor cells. And I don't have a light here. Oops. Well, can I have the light? No? No light? Okay, no problem. So This glioblastoma stem cell can give rise to the primary tumor, where you see different types of cells. You can differentiate, including stem cells. And the stem cells are the ones which stay behind after chemotherapy and surgery and so forth. And those are the ones under certain conditions which give, recurrence to tumor very quickly. But you see that the recurrent tumor has higher population of cancer stem cells. Okay, it's not that complicated as it looks like. Now the molecular oncologist, thank you so much, starts talking wireless. So our effort is to translate tumor engineering to the concepts of molecular communication. So if I isolate a cancer stem cell, we do that in the lab, and I put it in the proper microenvironment, we have some aspects, some language here of molecular communication. We have a living environment, which is the cell-extracellular matrix interaction. We have a communication medium, which is defined by different soluble ligands here. And of course, we have noise sources, where we have biochemical, thermal, and physical from pH, mechanical, electrical stimuli, because we need to put this under oxygen control, bioreactor, shear stress, and electrical simulation. So we are at the point where we can really start talking molecular communication, and environmental cues are present in spatially graded doses. So chemotaxis is the sensitivity to spatial gradient of solid chemical factors and ligands. Topotaxis refers to directed migration guided by the gradient of the nanoscale topographic features in the tumor's extracellular matrix microenvironment. So those two are already well captured here. What we need to progress further is haptotaxis, which is sensitivity to gradients of the physical chemical properties of cell adhesion, and durotaxis, much more advanced, which is sensitivity to gradients of substrate of stiffness and rigidity. And here we have a newly emerging field, mechanopathology. So we have major requirement for models recapitulating the three-dimensional topographic complexity and need multiple scales of size and organization. So we have now, going further, the glioblastoma stem cell give rise to endothelial cells, and I'm glad Professor Takano described that beautifully previously. So the glioma endothelial cell and glioma stem cell interaction is the epitome of complexity in chemotaxis and topotaxis spatial gradient. So tumor vessels can be recruited from glioblastoma stem cells. Glioma endothelial cells have a direct effect on distinct subpopulations of stem cells. So we hear, this interplay. And this orchestrates the activation of migratory, invasive, and other potential. The stem cells now can undergo to transdifferentiation, giving rise to functional endothelial cells. And we also have remote progenitors from the bone marrow generated endothelial cells. Therefore, there is a central, very central therapeutic challenge in glioblastomal management, how to tackle this particular interaction and how to interrupt it. The very first structural unit that arises from glioblastoma and endothelial cells is the so-called glioblastoma perivascular niche. That's where tumor recurrence arises and serves, I think, please correct me if I'm wrong, a prototype leader-follower based mobile molecular communication network. Why I think is that? Physical association and release of soluble factors via cell-cell interaction are augmented and amplified when we have those cells in close proximity. To functionally maintain cell renewal and stemness, the glioma stem cells require several autocrine and paracrine factors. Remember the previous slide, soluble and non-soluble signal molecules, not signaling, etc. Therefore, the perivascular niche, where we have endothelial cells and subtypes of stem cells, offers both. It helps the glioma stem cells to adhere to vascular structures where they can physically interact, and also allows access to increased localized concentration of soluble growth factors. And the physical proximity is therefore a key driver to tumor growth. So the perivascular niche evidently becomes a hub for multiple phenotypes, including self-renewal, maintenance, and angiogenesis. Therefore, we have a prototype system of natural bio-nanomachines. This subpopulation of glioma stem cells tethered to the vessel through chemotaxis, and another subpopulation tethers by haptotaxis, as we see here, in a more elaborated model. I believe that here we have a prototype model of application layer prototype functionalities for molecular communication application. So if you take the perivascular niche, this is the basal membrane of the vessel, endothelial cells, growth factors soluble, chemotaxis factors by this subpopulation here. So we have an enhanced gradient of soluble factors, and here we have migration of glioma stem cells directed by the physical tethering to the basement membrane. So the overarching functionality has to do with structure formation, where we have basic functionalities, bio-nanomachine location control through a chemokine as attracted for the endothelial cells, and environmental monitoring, so have monitoring of the molecular communication environment, distribution and migration towards the laminin, which is here, and molecular concentration control, and this we can achieve with delivery of growth factors through nanocarriers. And of course we have support at the physical layer, since the glioma stem cells serve as mobile bio-nanomachines, and we have detected concentrations of growth factors, tumor growth factor beta, for example, which is released by the endothelial cells here. And we have moved migration along the gradient of laminin, which has the basal membrane of the endothelium. I mentioned that the key point here, that is very important to have nanocarriers for growth factor delivery. The local presence of growth factors and tumor progression biomarkers at the tumor site is very, very significant. However, there are major drawbacks if we give them directly as solubles in the culture, in the three-dimensional models. There is short biological half-life, rapid degradation, toxicity, and poor passage through biological barriers. So it's important to use them as we use drug within the nanocarriers, and they have similar activity as we do when we study drug delivery. So the required functionalities here is bio-nanomachine location control. In order to have distribution of the growth factor carrier bio-nanomachine, this can be metallic nanoparticle, a liposome carrying NGF, FGF, etc., and they indicate a cell niche within the tumor model. Need to have environmental monitoring in order to sense the concentration of the growth factors and have a concentration of the molecules guiding the localization of the bio-nanomachine, as we have with the tumor growth factor beta. Monitor the molecular communication microenvironment and have molecular concentration control. For example, growth factor concentration at the target site, as we need for vascular endothelial growth factor, and guide the ligand concentration in the environment. And this is an example of some work in our lab, where we have magnetic nanoparticles loaded with growth factors within the nanomembranes we use as scaffolds for three-dimensional tumor growth. So, putting all this together, I come to say that the perivascular niche and glioma stem cell, glioma endothelial cell interbrain can function as an externally controlled molecular communication network, where we can intervene in order to have therapeutic applications. So, and I thank, Professor Cecano for this, very well organized model of externally controlled molecular communication, where the overarching functionality is multi-scale in messaging. So, what we have to do here, as biologists and physicians, is have transmission of information from bio-nanomachines to external device and vice versa. Thank God, Kostas, for talking about building interfaces here. So, what we try to develop is platforms of genetically modified biosensors functioning as transmitted and receivers bio-nanomachines, optically active, in order to get quantitative data on the spatial distribution of these biosensors and have validated protein interaction networks recapitulating communication processes in the microenvironment. And the very important thing is to identify the best combination of donor and acceptor if we have optically active nanomachines, in order to have the best combination and have a complete network here. And these are some suggestions we already tried in the lab. I mean, DLL as energy donor, and notch receptor as energy acceptor, IL-8 as donor, and CXCR1 receptor on the stem cells as energy acceptor, and so forth. And most importantly, integrin alpha as acceptor here, which can have a combination of diffusive and adhesive molecules at the same time. And finally, quantitative and qualitative analysis of the spatial temporal distribution in real time in vivo. And I did some help here. I know what to measure, but I don't know how. So we are in search of the suitable hardware. Very quickly, different other opportunities, of interfaces which are provided to us, through the different systems of nanoparticle drug delivery we have here, okay, and the environmental responses drug delivery can serve also as interfaces in order to have controllable, molecular communication systems, and the good detection and the good selection of the active targeting strategy, and thank God glioblastoma provides multiple. And finally, of mice and men. Can we have preclinical translation of molecular communication systems? Where can we go? How close are we in order to maintain reproducible interfaces in the living animal? In vivo electroporation of tumor xenografts, it's a very powerful technique. Electroporation will create holes on the cell membrane in order to introduce material based on the electric charges. The nucleus and the mitochondria, for example, are very negatively charged. And the technique is very well developed already, and we can achieve beautiful results, with in vivo electroporation in the rodent brain. And now we also have external tweezers that allow us, with non-invasive technique, to have electroporation of several genes at the same time, and with high reproducibility. So this way we can have sustained systems of glioblastoma and Beyeric mice expressing at least one pair of transmitter-receiver optically active molecules and eliciting reproducible and quantifiable threat signals. But most importantly, need to select adhesive molecules for transfections, like laminin and integrins. Why? They have advantages over diffusive molecules. The attractor concentration is not impacted by flow in the environment, and the attractor concentration remains high for a long period. Therefore, we have accurate detection of the concentration of the bio-nanomachines. And what this translational nanomedicine I'll be talking about is nothing else but a new cliche for an old tale, from the bench to the bedside. So years ago, when I was a postdoc, basic researchers were working alone, clinical researchers and those with applications in another part of the world. And the companies and therefore the technology transfer, preclinical studies were done remotely, and none was knowing what was going on. What we need here is from the very beginning to have basic researchers, translational research, patient samples, clinical research and practice all together in a team. How can we do it? Can we move towards translational nanonetworks and translational molecular communication? Or are we lost in translation? We have to admit that Europe is lagging much behind US and Far East. But hopefully, with circle activities and opportunities, and with clear vision, we can achieve a glorious victory, like the victory of Samothrace, which is not in Greece, but at the Louvre. And, next time we have much more to present. And now I want to thank, people who have stimulated all this, people, who have or are still working in my laboratory. And of course, my colleagues at the Department of Engineering, University of Cyprus, okay? Kostas Petris, who is here, Dr. Stylianopoulos, who is doing mechanopathology, and of course, Dr. Krikidis, who has led my way to molecular communication. And I feel very excited at this stage of my career, going through a brave new world. And thank you, all of you. Okay, let allow the younger people to talk first. Yeah.
Thank you for your talk. First question: are these glioma endothelial cells are much different from other endothelial cells, or are they originating from cancer cells, or just for usual endothelial stem cells? So, how much they are different from usual endothelium? And another: do I understand right that you co-cultivate them with glioma stem cells? And if so, do these endothelial cells form like a vascular, or just they spread out without forming tubes and so on?
Well, actually, I don't know if I can go backwards back. Okay. These are the vessels formed by tumor endothelial cells, and these are the vessels formed by normal endothelial cells. Now you see the major difference. The vessels arising from tumor endothelial cells, which arise from transdifferentiation of glioma stem cells, are very fragile, very disorganized. They don't have basal membrane. Yeah, it's a total indifferent phenotype. Okay? All right. So what's your other question?
Can you do the structure like in vitro in some models, as you have shown with layered things? So do you get then this kind of phenotype of vessels, or you just have the endothelial cells?
Okay, all right. So far we have been able to have early structures, of vessels, accumulation, what we saw that transmigration. Okay, but alone the glioma stem cell cannot give the overall structure. Here we need microfluidics. We can count to a certain point, but after that we have to call in microfluidics expert, But starting from glioma stem cells, you can have a much better and faster, model recapitulating the vivo situation, but you cannot have delivery through all the way through. You really need help from microfluidics. All right. If nothing else, I really want to thank all of you for staying, up here, and all the speakers. I'm sure you agree with me, except me, they have given all exciting, stimulating presentations. And I hope, that the younger generation is able to take forward, a little way. Thanks a lot.
Coffee outside, but we don't have much time, so maybe 10 or 15 minutes, because there's a hard stop in here at 5 o'clock after the next.